All right, so we're going to start on Chapter 10 today. Chapter 10 is genetics. We're really going to be in Chapter 10 for pretty much the rest of the semester. So there's a lot of different things that we're going to talk about throughout this chapter. So today is just a general introduction to some of the terms mostly. This is really a kind of a vocab class. We're going to be getting into a whole bunch of kind of example problem sets. I'm going to hand you out at some point today a worksheet that we'll work on probably in class on Friday if we don't have time today. We'll see how much time we have. And then we're going to start working on harder and harder problems as well as adding in different kind of concepts about genetics. And that's really what Chapter 10 is all about. So there's going to be a lot of problem sets. I'm going to post all of these on Canvas as well so you can download other copies of them or if you miss getting a copy or something like that. So just before we get into what is genetics, just going to review a little bit some of the concepts that we've been learning over the last two chapters that lead us into genetics. So chromosomes, which are what all these little lines are here and what the big X's were that we were separating during mitosis and meiosis, are packets of genetic information. So DNA is wound into chromosomes by wrapping DNA around histones. Chromosome is a packet of DNA. That DNA on a chromosome contains many, many genes. In most human cells, there are 23 pairs of these. Again, that number varies. And we know that we go from having a diploid cell where we have paired chromosomes to a haploid cell where we have single chromosomes during meiosis. And then when two haploid cells, sperm and egg cells, come together during fertilization, then you get a diploid cell again. So, in genetics, we're going to be looking at chromosomes, but we won't really be talking about them as chromosomes for most of the chapter, as we're going to be looking at specific genes on these chromosomes. I'm going to get to that in a minute. Chromosomes in a diploid cell, so in the cells that are in, most of your cells that are in your body, are paired. Each autosome is a member of a homologous pair of chromosomes. So this would be one autosome. This would be its homologous pair. So you have two pairs. Members of a homologous pair have the same genes, but might have different versions of those genes. And we call different versions of the same gene alleles. So if you hear the word allele, that's what it means. It means it's a particular version of a gene. So for instance, there's a gene that controls hair color. You can have the blonde version of that gene. You can have the brown version of that gene. You could have the red version of that gene. Those are different alleles of the hair color gene. So gene is a packet of DNA that codes for a specific trait, specific protein, and an allele is a different form of that gene. So homologous pairs, again, remember, what we mean by homologous pairs of chromosomes is that they have the same genes on them, but they could, again, have different ones. So, in a diploid cell, how many copies or how many alleles for each gene do you have? Two, right. There we go. Pretty simple. So diploid cells have two alleles for each gene. Ignoring more complicated things that actually have multiple genes coding for them. But you have two alleles for each specific gene. The two alleles can therefore be identical or they can be different. So you have two homologous chromosomes. You could have identical copies. So here on this graphic, the A gene here has a capital A on the pink gene, chromosome and a capital A on the blue chromosome. So they'd be identical. They could also be different. Here we have a capital B on the pink chromosome, but a lowercase b on the blue chromosome. So, and what we're going to be kind of talking about throughout today is what effect does that have on an organism, first off? So having two of the same versus having two different. So two of the same, you're pretty sure what that individual is going to look like, because if you know what that capital A gene codes for, they're going to be expressing that capital A gene. But what's going to happen in this individual, this gene that has two different forms, a capital B and a lowercase b? That's what we're going to talk about today. And then we're going to talk about how does this individual having two of the same or two different affect the likelihood of what's going to be expressed in their offspring. So we're going to look at this individual versus another, combined with another individual, and what happens to their offspring. So a lot of this work started with Gregor Mendel. So Gregor Mendel was lived in the 1800s, and he did a lot of his work in the kind of mid 1800s or so, mid to late 1800s. He has a really interesting life story, and if you have a chance to read about him, it's really kind of an interesting story. His family were very, very poor. They were a sharecropping family, and apparently Mendel was really awful at farming. He was not at all good at farming. So he couldn't help his family to grow food and couldn't help in the family business. So what they did is they sent him off to be a teacher. Basically, that's what you did with people that couldn't do anything useful. Sent them off to teach, right? So only hurts a little bit, right? But he was prone to depression, and he was actually sent to a location where they didn't speak the same language as him. So he was a pretty awful teacher, considering he spoke a different language than all of his students. It was hard for him to have any success, and he was pretty depressed about it. And the teachers at the time, again, were monks. It was the monasteries that did most of the teaching at that time. So because he had trouble teaching, he wasn't teaching all the time. The abbot at the monastery sent him to work in the garden, and it was a very progressive kind of monastery that was really into science. And his kind of task was to learn how to breed better plants. And he actually, kind of went full circle, went back to almost farming again, and learning how to breed better plants. And what he worked on, at least what he worked on and wrote a lot about, and what we've learned about post-Mendel, is his work on pea plants. And what he would do, so a pea plant has kind of a very useful for genetics characteristic that it can self-pollinate. So you can take the pollen from a pea plant and you can fertilize itself and do a self-cross. And what he would do is he would do really kind of careful transfers, either by self-pollinated plants by hand or by taking pollen from one pea plant and putting it in the flower of the other pea plant to cross them. He would actually remove parts of the flower so that they wouldn't do crosses that he didn't intend. He was very, very careful, very meticulous about this. So he would take pollen from one plant, put it onto another plant, grow up the seed pods. He would then plant the seeds, and then he would grow them, and he'd record everything that happened. And he'd record all the characteristics of these plants. There were many different characteristics he looked at, including plant height, flower position, plant color, seed shape, all kinds of things. And we'll talk about some of them throughout this chapter, as he did. I was very meticulous about them. Kind of the downside of peas, although they can self-pollinate, the downside is that they take a very long time to do this. So he basically do pretty much one cross a year, but he'd be doing this on thousands and thousands of plants. So he'd be doing many crosses, but he can only do one generation a year. So Mendel learned a ton of interesting things and a lot of stuff that revolutionized genetics, but we've learned a lot more since because we've picked new organisms, and I'll talk about that later on in this chapter. What he was looking at is the different combinations of offspring, and he kept really good records of exactly who the parents were and exactly who the offspring were. So he would do self-fertilization and self-crossing of different combinations and use that to deduce or to figure out ideas about inheritance. So he would take a green pea plant and he would cross it with itself, and he'd find that the offspring were all green. Or he'd take a plant that grew from a yellow seed, cross it with itself, and find out that the offspring were all yellow. And then he'd find interesting kind of differences. So those were things he might have expected. Self-cross would produce individuals of the same type. This is what's called true breeding, or a purebred or true breeding line. This is often what he would start with. He would make sure that he had these true breeding lines that he knew every time they cross them with themselves, all of their offspring look exactly like them. And that's a true breeding line, and that kind of gives him kind of this pure genetic traits to start with. But then he would find that he would have hybrids where he would cross them with themselves, and he wouldn't get all individuals that looked like the parent. He would get some that were different. So here's a yellow crossed with a yellow, and he ends up with 3 yellow seeds and 1 green seed here. So we get these interesting differences. And by recording all those differences, he was able to come up with a whole bunch of ideas about how genetics worked. And kind of the really cool thing about his work is a lot of it was done in kind of the 1860s, 1880s, kind of that era. And we didn't know about DNA, and we definitely didn't know DNA was a genetic material until the 1950s. So this is long before any ideas about DNA. So we are going to apply concept that we learned in mitosis and meiosis on this process because we know about that now. But Mendel's time, he knew nothing about it. Mendel was actually pretty obscure at his time. He did write kind of 2 big papers and tried to present them, but again, he was nervous, prone to depression, a horrible public speaker, so he was talking about crosses and ratios and stuff like that, and the whole audience fell asleep, so no one knew what he did. Until about 20 years after he died, just after 1900, when 3 different labs across the globe all tried to plagiarize him at the same time. And then they sent each other their papers and said, "Oops, you found the same obscure paper that we did," and copied it. So there was a rebirth. So Mendel didn't get credit until after he was dead for all his work. So we are going to talk about some of the stuff that he came up with. And then later on in this chapter, we're going to talk about some things that are different from Mendelian genetics, from the basics that he kind of solidified, and learn how things get a little more complex the more we learn about genetics. So, before we fully understand what's going on in these crosses, we need to know a little bit of the terminology that is associated with genetics. So, we'll use the word dominant and the word recessive a lot in genetics. And unfortunately, in the English language, the word dominant has taken on a different meaning than we use it in genetics for. The word dominant often means most common in English language. In genetics, dominant does not mean most common. Dominant is a very specific characteristic of an allele. A dominant allele exerts its effects whenever it's present. So whenever a dominant allele is present in an individual, you're going to see the results of it. So crossing a yellow seed plant with a green seed plant always yields yellow seeds. This tells us that yellow seed is dominant. So you can cross a yellow and a green and get all yellow. You can cross a yellow and a green and get some yellow and some green, but you're always going to get some yellow. This tells us that whenever that yellow gene, whenever that yellow allele is present in an individual, that individual is going to be yellow. And that's what dominant means. Doesn't mean that it's more common than green. Green can be way more common, and we'll talk about cases of dominant alleles that are actually fairly rare in population. But dominant means that if it's there, it's expressed. So the opposite of dominant is recessive. And a recessive allele is one whose effect can be masked, and is masked if a dominant allele is also present. So in this case, this true breeding green plant might have passed on the green allele to its offspring, but because there's also a yellow allele, these offspring are yellow and they're not green. So a recessive allele can be masked. And we often say when a recessive allele is present but it's not expressed, we say it's being carried. So these yellow seeds could be carriers of the green allele, but they're not expressing it. So a carrier is something that has that gene present, has that allele present, but is not expressing it. Okay, so recessive alleles usually encode non-functional proteins. So think of it this way. Let's say you've got a flower, and you've got a flower that either has a white color or a red color. Let's say this flower has 2 copies of the red gene. They're going to be red, right? If we have a flower that has 2 non-functional genes, maybe the default is white. But if they have a copy of the red and they have a non-functional version, what color are they going to be? They're going to be red, because they're only expressing red. So this is often what happens in recessive genes, is that they're actually non-functional versions. And so the default color in this P might be green, and the yellow, when it's expressed, covers up that green coloration that's masked. So the green allele is actually non-functional. It's not producing green pigment, it's producing no pigment, and kind of the background is green here. Okay, and we'll get more into that. Okay, so the question Mendel had, so he could see things like this. He said, well, if yellow seeds are dominant, why are some green seeds, or some seeds green when a yellow seeded plant is crossed with a green seeded plant? So in order to understand that, we need to know a little bit more information before we can answer that question. So it makes sense that green crossed with green gives you green, yellow crossed with yellow gives you green, and maybe yellow crossed with green gives you yellow if yellow is dominant. But why can you have yellow crossed with green give you green and yellow? That's what we're going to get into next. Okay, so individuals have 2 alleles because, for each gene, because they have 2 homologous chromosomes, right? So this is what I kind of started drawing here. You can have 2 copies of the dominant allele. So in this case, red here, or yellow. So up here we're saying yellow, we're going to use the letter G. Oftentimes a dominant form of a gene, a dominant allele, will use a capital letter. So homozygous dominant is an individual that has 2 dominant alleles for a gene. This would be big G here. 2 copies of the yellow gene. You can be homozygous recessive, which means you have 2 copies of the recessive allele, so little g. Or you can be heterozygous. A heterozygous individual has 1 copy of the dominant and 1 copy of the recessive allele here. But basically, the word heterozygous, because we'll get into more complex thing, heterozygous means you have 2 different alleles. Homozygous means 2 of the same. So homo means same, hetero means different, homozygous 2 of the same. You can have 2 of the same dominant, 2 of the same recessive, or you can be heterozygous. You can have 2 different. Right? So those are your 3 real options here, especially since I've only given you 2 alleles. So just a little bit of nomenclature on how we're going to be writing these things. For one gene, we pick 1 letter. So you can pick any letter you want. 1 gene gets 1 letter. So here, the seed color gene gets the letter G. But you can have different versions of that letter. The capital G equals the yellow allele. The lowercase g equals the green allele. So a heterozygote has 1 of each allele. Right? And we'll get more into that when we get into problem sets. Okay, so here's a dominant recessive question to see if you can figure this out. Okay, in Europe, light hair is common in Northern Europe, and dark hair is common in Southern Europe. So which of these following statements do you think is most likely to be correct? All right, you guys pretty confident? Well, I'm getting there. Well, 80% of you have chosen one of the answers. All right, I'll show you what you guys have said. I hope it's 83% now. So most of you have said D. D is wrong. Okay, so what does dominant mean? means if it's there, it's expressed, right? And recessive means it can be masked, that it's not always expressed. Do you think if dark hair was dominant to light hair, that it could be different in other parts of the world? No. So let's say, like I was saying, most recessive alleles are because they're non-functional. So do you think just because you live in Southern Europe that the expression pattern would change? So if you go from Northern Europe to Southern Europe, is your hair color going to change? Right? Not unless you get it dyed, right? So the dominant versus recessive. This is a big misunderstanding, which is why I put this question up here. Dominant does not mean most common. Recessive does not equal least common. They're expression patterns. Their dominant means if it's there, it's expressed. So given the information I gave you, you have no clue at all about dominant and recessive. All you know is that light hair is common in Northern Europe and dark hair is common in Southern Europe. So if light hair is common, how common do you think the allele for light hair is? Is it common or rare? If light hair is common, is the light hair gene common? Yeah. If dark hair was common, would the dark hair gene be common? Yeah. So that's really all you know. So these ones were tricky. They threw you off because it played on your desire to use the word dominant as common. Dominant does not equal common. So dark hair alleles are more common than light hair alleles in Southern Europe, but not in Northern Europe. That would be the right answer, right? So only a few of you got it, but I deliberately tried to throw you off here. So be careful. Dominant does not mean common. The dominant hierarchy cannot change by going to a different part of the world, which is why if you move, your appearance doesn't instantly change because you're in a different part of the world, right? You're going to be expressing things in the same way no matter where you are. It's just that the instance of that, or the commonality of those particular alleles might change in different parts of the world, which is why expression changes. Okay. Okay, so all right. So dominant things are expressed whenever they're present. Recessive is masked in a heterozygote. You can have homozygous dominant, homozygous recessive, heterozygous. So now, how do we figure out the genotype of the parent? I'm going to tell you what the word genotype is, hopefully, in a minute. Yes, next slide. So we'll get into that. We're going to use something called a Punnett square to help us determine what's going on in these crosses. Why in this cross did we cross a yellow and a green and get all yellow, whereas in this cross we crossed a yellow and a green and got half and half? Well, what turns out to be the reason is that this yellow was homozygous dominant, whereas this yellow was heterozygous. So big G, and big G, little g, both express yellow, because yellow is present in both of them, but we're going to see that they have a different ability of passing on traits to their offspring, and a heterozygous individual has the possibility of passing on that little gene, the little g there. So we're going to talk about that. So whenever we do genetic problems, I'm going to ask you for one or both of these things. So knowing what these are and the difference between them is pretty important because that'll be the answer to most of these questions. Genotype is a description of the two alleles of an individual. So the genotypes we have here are big G, big G, big G, little g, and little g, little g. So here, genotype equals those three options in this case. Phenotype is what's expressed. It's a description of the trait an individual expresses. So in that case, it would be yellow or green. Okay. And I'm going to be asking you for ratios of these things. So if I ask you for the genotypic ratio, you're going to tell me the number of homozygous dominant to the number of heterozygous to the number of homozygous recessive. If I ask you for the phenotype ratio, you tell me the number of yellow to the number of green. And we'll get to that as we go along. Okay, so because homozygous dominant and heterozygous both give us yellow individuals, these two genotypes give you the same phenotype. So oftentimes there are many different genotypes to give you the same phenotype. And this is why your ratios, your genotypic and your phenotypic ratios, might be different. All right. So how do we know what's going on? What I'm going to do is, while I'm talking about this, I'm going to pass around these. So just, thank you. All right. In order to determine what's going on and to really have a good understanding of how genes pass from parent to offspring and how what the parent looks like can affect what the child looks like, we're going to use something called a Punnett square. A Punnett square uses the genotype of the parents to reveal which alleles the offspring may inherit. So, here are two parents that are heterozygous. So they have one big G and one little g. So their phenotype, because we know yellow is dominant to green, is going to be yellow. If an individual is big G, little g, they can either pass on a big G or they can pass on a little g to their offspring. And this is directly related to meiosis. So during meiosis, let's say this blue marker is the big G and the red marker is the little g. You've got a chromosome with the big G on it and you've got a chromosome with the little g on it. They're going to pair up in meiosis 1 and split so that the egg or the sperm have a 50-50 chance of either getting the big G or getting the little g. So the female gametes, the egg, will either have a big G or they'll have a little g. And same with the sperm. The male was big G, little g. He's going to either pass on the big G or he's going to pass on the little g. So to figure out what the potential offspring are, we need to see what potential combinations we can get. So the male could give a big G and the female could give a big G, in which case we'd have a big G offspring, which would be yellow. The male could give a big G and the female could give a little g, in which case we'd have a big G, little g, and we'd have a yellow heterozygote offspring. The male could give the little g and the female the big G, in which case we'd get a big G, little g again, yellow offspring. And then the final possibility, both parents could give their offspring a little g, in which case we'd have little g, which would be homozygous recessive and would be green. So in this case, the genotypic ratio would be one way of getting big G, to two ways of getting big G, to one way of getting little g. And the phenotypic ratio of the offspring, there's three ways of getting yellow to one green. So when I'm asking for these for a test, thank you, I want you to write them out like this. So one big G to two big G to one little g, or phenotypically 3 yellow to 1 green. Okay, so this is called a Punnett square, and all right, let me see. Okay, let's practice some of these. So I just handed out a big set of worksheets. So start with this front page and work your way through these. And what I want you to do is do a Punnett square for the parents that it says, and give me the genotypic ratio and the phenotypic ratio for each of these. And I'll be walking around to help you as you do it. So just to do one of these maybe as a class, let me just do this for a second. And just to show you, those of you that are, this has been really straightforward for you to make it a little bit easier, and this will set us up for the next one. So if I do problem 1, a green pea plant, big G, little g, is crossed with a yellow pea plant, little g. This might seem excessive right now, but we're going to get to more complicated problems. It helps to have a system to set these things up. What I like to do is I like to write out kind of a key for myself before doing any of these. And I know none of you probably did this. You just barrel right ahead and did it. And it's fine because these are easy problems. They should be. But I often like to write down the phenotypes and the letters that go with them. So here I know I have green and I have yellow. So I have green and yellow. And in this case, green is represented by a capital G and yellow by a lowercase g. And that just helps me in the future when I'm trying to analyze these things. This will especially be helpful on the back side where I didn't give you letters in the questions. So a green pea plant, big little g, is crossed with a yellow, which is little g. I like to write out the parents as well, so I have that here. So this parent can give me either a big G or a little g, so I'd put that on one side of my Punnett square. This parent, what can it give me? Only little g's, right? So you could, which probably all of you did, write out 2 little g's, but if it can only give you one gamete, you don't need to write it twice. So you can skip over that. So if I was to write out the gametes here, I have big G and little g, and here I have little g and little g. Because both of these are the same, you can actually get rid of the second one, and that's fine. So I could do a Punnett square that just looked like this, in which case I'd get a 1-to-1 ratio for both of my things, which is the same as a 2-to-2 ratio. So this actually lets you cut it down to the common final. If that doesn't make any sense to you, by all means, write out the full Punnett square. You're going to get the same right answer, and that's fine. So those of you that understood what I just did, we're going to use this again when we get to more complicated problems, and it's going to save you a lot of time when you're writing 16 squares to be able to cut it down to 4, like that. So here I'd have my genotypic ratio of 2 big G little g to 2 little g, or 1-to-1. And I have my phenotypic ratio of 2 green to 2 yellow. Okay. So, all right, we'll do some of the ones on the back side of that on Friday, and we'll kind of go on then to more complicated things. So the whole idea here is to use Punnett squares to tell you something about the offspring. It also can use Punnett squares to tell you something about the parents, and we'll talk about that on Monday or on Friday as well. So I keep hitting the same button over and over again. That's not helpful. All right. So we'll leave it there because it's about the end of class. If you want to work on these on your own, you're welcome to. We'll do a few more problems on next class.